Dr. Hitendra M. Patel from the Department of Chemistry, Sardar Patel University, Gujarat. And he'll be speaking on his topic entitled as Synthesis of Small Heterocyclic Scaffolds Enabling Anti-Cancer and Anti-Glioma Cell Line Targets.
Respected chairperson and the speakers, research scholars and students, ladies and gentlemen. So first of all, I would like to thanks to the organizers for this beautiful congress. Friends, I will discuss my work with you based on the synthesis of small heterocyclic scaffolds enabling the anti-cancer as well as the glioma cell line studies. So friends, here I would like to show you the outline of my today's talk. I would like to discuss, please give me the microphone. Hello. I will talk on the introductory part of my synthesis methodology as well as the multi-component reactions of the organic chemistry by making the cyclizations with the formation of a CC-CN bond and CO bond formation for the three important scaffolds. And here it is, you can see here, the 3,4-dihydroxypyrimidones, another derivatives, 6-amino-2-pyridone, 3,5-dicarbonitrile, and the third one, that is pyreno-2,3-C-pyrazole. So these are the three important scaffolds we synthesize with their libraries, and we check the anti-proliferative activities as well as the glioma cell line studies. Well, if I'm talking about these three important the derivatives we synthesize by using the multi-component reactions, and we know the importance of the multi-component reactions that will reducing the steps. As we know, the total synthesis of the drug molecule, it takes huge time and the cost effective and so many factors there. So that's why it is benefited. So let me try to clarify first. I'm a simply organic chemist rather than medicinal chemistry. So hope you can understand my presentation. And I choose that the synthesis based on the ionic liquid use, deputatic solvents, and the other Lewis acid based catalysts, and the some of the organocatalysts, we use it for synthesizing the kinds of derivatives. Well, I would like to discuss some of the introductory part of this glioma cell lines. What do we mean by the glioma cell lines that you will find here? As we know, glioma cell lines, the glioma is a growth of a cells that starts in the brain and the spinal cords. And the cells in the glioma look similar to the healthy brain cells, and it is called the glioma cell lines. When the glioma cell present surrounding the nervous cell and help them to function, but when the glioma grow and it forms a mass, then it is called the cell lines. And it is very early to diagnosis as the causes, right? So, and we have seen that the literature also and so many things, within the 1 year, if it is diagnosed, then the patient will die within the 1 year after diagnosis of the causes. Well, based on the literature survey, whatever the molecules we identified for the potency of the glioma cell line, that you will see here, pyridone core with the cyano groups, as well as the pyridine core with having amino and cyano groups. So these are the key functionality for this core for having a good important activity as anti-glioma cell lines. You can see here the good the value that can be quoted here with the different researchers. So this is one derivative I am talking about. We have synthesized a one-pot reactions for the 6-amino-2-pyridone 3,5-dicarbonitriles. So with using 2 steps, we synthesize this core as a pyridone. And here we have some library, and one of the molecule which shows the greater efficacy as a 5O molecule with having effective concentration that is 15 micromolar. And we are targeting these 4 different cell lines. And friends, here you will find whatever the existing drug we are having into the market, we did the combination approach also with the existing drug molecules, that kind of study which is involved over here, and you will find this. I will not discuss much more regarding the path for the synthesis of this one. As we know, it's a World Cancer Congress. So friends, we characterize all this data with using the spectroscopic analysis as well as the single crystal grow. We develop the single crystals and then we characterize this molecule. So let me try to explore the cytotoxicity activity of the synthesized molecules with the diverse cancer cell lines. We use the six different solid cancer cell lines. Friends, we are receiving the data from our collaborators from the Dundee University, UK, Scotland, and another one from the Spain, Josem Padron, who helped us regarding these kind of studies. And I'm able to hear that is only based on this collaboration. Otherwise, you know, I'm a simply organic chemist. So friends, let me try to explore over here one of the 5O compound which shows the greater efficacy here, and you will see whatever the drug molecule which is available. So dark one, it clearly indicates that this is our simplest molecule which shows the activity against the cell line. So you will see here the relative viability. Dark one, this is the 5O molecule, and I'm talking about this is the simplest drug which is available into the market, and the graph you can see. And when we have to go for the combination of our 5O molecule with the existing drug molecules, then you will see that the better efficacy we can have it with the combinative studies. So that is the finding we did it here with the help of collaborators. So you will find here the C is nothing but the combinative approach. We can have a good results. I will not discuss much more regarding this, but you can see the cell lines which is coded GL261 cell were treated with the 5O alone, and you will see that the kinds of the activities. Now here, friends, you will find the in vitro anti-glioma activity. When we talk about the best finding we have with the 5O molecule, that is 15.1 effective concentration that can be measured with this GL261 cell lines. And this work is published over here. That there is a huge docking and other relating that the things, ADME tools is also there, but I didn't include it here. Now, if I'm talking about the another types of molecules, so friends, another core you will see here, that is a pyrazole cores. In this pyrazole course, many of the scientists, they claim as anti-proliferative activity as well as the glioma cell lines. And here you can see that the pyran core, these are the two important core which is very essential to show that the great potency. So based on that literature survey, we designed this molecule with respect to the pyrano 2,3-C pyrazole molecules, which enables the PKB beta as well as AKT2 inhibitory activity as well as the in vitro anti-glioma activities. So friends, we have certain libraries with having this pyrazole core with having the cyano group as well as the NH2, and we did some of the 2D primary cell study with the 2D primary cell lines as well as the 3D neurosphere with these molecules. So here the two molecules we identified as a great potency. So I will not discuss that the chemistry part, how we can synthesize it, but the DEPCO is the catalyst, we use it, and as far as the green solvent system is concerned, ethanol we use as a solvent, and within 30 minutes we are able to synthesize this one. So there is nothing but we are focusing the sustainable chemistry also for designing a new heterocyclic molecules. So I will not discuss the chemistry part. If I am talking about the in vitro anti-glioma activity, then here 4J compound, according to the published work in the journal, the 4J compound which found out the great potency and 20 plus and minus EC50 value with the GL261 cell line. So, and we also found out that the in vitro kinase inhibitory activity which shows the desirable activity with the 4J compounds. So as you know, the serine or the threonine kinase is called the AKT, and it is also known as the protein kinase B, PKB beta inhibitors. So here our one of the molecule that is screened, I mean the 5 micromolar compound of a 5J that can be screened with the 139 purified kinase screening, and we found out here the IC50 concentration IC50 value of this with this AKT2 as well as PKB beta is found out 12 micromolar. Whereas in this case we identify the AKT1 target with the IC50 value 14 micromolars. So, and then later on, the AKT2 is overexpressed in the low-grade glioma, and the primary expressed in malignant cell line studies is also identified. So, establishing cytotoxicity of this compound is very essential, and my collaborator did this, and you will find here the 4J compound with the 0. Red one, it indicates that the 0 micromolar, blue is a 10, and here it is a 30 and 100. According to that, you will see that the 4J molecule with the different concentration, we have checked the cytotoxicity with respect to the GL261, GMB6, GBM6 as well as GBM22, and we found out that the good desirable activity, desirable cytotoxicity. And when we send our paper to the Annals of Medicine, then the query arises, and that query will be solved by my collaborators, and they ask us to check the cytotoxicity activity with the normal cell lines, and the normal cell line activities which is shown here, and it is a desirable one. And you will find these publications with the Annals of Medicine journal from the Taylor and Francis. Now, another third scaffold, if I would like to mention you here, there is a monster analog. So friends, we are knowing very well the monster analog series of drug. It is a well-established drug, and it is available into the market with having, I'm talking about the pyrimidine core, pyrimidone core. With having this one. And as you know, if I'm talking about the OHS group is there on the meta position with respect to this monster analog. But we have to, based on the existing drug molecule, we designed this such a library with inserting this molecule in ortho, meta, and para, and comparative study we also carried out. So the 20 examples we synthesize in our molecules with the new molecules, and we check the anti-proliferative activity as well with the 6 solid tumor cell lines, as well as we identified one of the molecule as a 4Q. It shows the growth of inhibition 50 value, GI 50 value, that is 1.1 to 3.1 micromolar. And the ADME predictions through the computational approach also we did it, and it found out that the drug likeness properties and the physiochemical properties of the drug likeness properties is also carried out. And this work you will find with the ArcFarm journal. So here, monster analog, according to the literature survey, you will find here the OH group is present on this third position. So we just simply modified this one, and we want to check it whether with the ortho, meta, and para, if we have to take another substituents, then what will happen according to the comparison with this existing drug molecule as a monster analog. So monster analog, that is nothing but I'm talking about it is a EG5 kinase, the target. So essential kinase for the cell division with the EG5 that you will identify. And many of the scientists, they reported this work based on these kinds of activity with having this molecule. So here, based on the literature survey, we designed our molecule by taking these 3 important molecules in a multi-component reactions with using the lanthanum triplet and ethanol as a solvent at 100 degrees centigrade. We synthesize the new derivatives with respect to the pyridone derivatives. So there is nothing but I will not discuss the optimization reactions and with the what type of percentage yield that we identified. So we develop a single crystals in our laboratory, and we check it, the sorted diagrams you will find here, and it is confirmed that whatever the molecules we synthesize, that is satisfied with each and every concept of these molecules, C bond, C N bond, and other pseudochemistry that can be identified. Another thing, if I would like to explore this library of these monster analogs, the anti-proliferative activity of this, okay. Sorry, I'd like to conclude it here. This is the anti-proliferative activity. Just one thing I would like to mention you that as comparison, where the functionality of the OH and the functionality of the O and C6H5, you will see here, this is the last slide I'm talking about, orthobenzyloxyphenyl dihydropyrimidones, meta position and the para position. And we identified here, so this para position, if we can have a benzyloxy, then it shows the wonderful activity with respect to these 2 important molecules. We find this drug molecule, and the other study you will find it from this journal. So friends, as you know, these are the 3 core. I claim here one of the molecule that shows the good activity, and based on that we have to have a comparison with the existing drug molecule. And here also the 2 compound which shows the good the targets. And here also 1.1 to 3 point something micromolar, the potent activity is found out with one of the core with having the para positions. So I will not discuss this one. So these are the conclusions, and these are our publications in last year. There may be the missing many. These are the reference and acknowledgement. I am from the Sardar Patel University, Hitendra Patel. My collaborators is, I'm here that is only based on this Josem Padron from the Spain, Saurav Banerjee from the Dundee, University of Dundee, and the other collaborators. This is my recent team. One is already completed here, and the majority of the work is shown here by the Rutu Raj Singh here. And special thanks to the organizers and for funding SCRB and our university, and I'm associated with the Royal Society of Chemistry as an elected member and the fellow also. So thank you very much for your patience. Thank you very much.